how does it work and why it is useful. Let us dive in and learn more about SDS page. SDS page stands for sodium dodecyl polyacrylamide gel electrophoresis, and it is a technique that is used to separate protein based on their size, and it is one of the most widely used type of protein electrophoresis out there. Electrophoresis is a standard laboratory technique which charged protein molecules are transported through a solvent by an electrical field. Biological molecules tend to carry a net charge at any pH except their isoelectric point, meaning that they will move to the positive charge of an electric field. The mobility of a molecule through an electric field will depend on electric field strength, the net charge on the molecules, as well as the molecule size shape, and lastly the properties of the matrix.
In this principle, the electrically charged particles or molecules in the electrolyte medium migrate to the electrode with the opposite sign when the electric field is applied in a vertical SDS page system. When the protein samples are loaded into the well, the proteins will be stacked in the stacking gel before entering the resolving gel. When the electric field is applied, the proteins begin to migrate from negative terminal to positive terminal at the same time as the sample migration direction shown in this figure. Then the proteins will be separated according to their molecular weight in the resolving gel by being presented with bands. In this case, the particles or molecules flow at different rates depending on their molecular weight and charge of the protein sample. Apart from that, larger molecular weight proteins tend to move slowly through the geometry, and small molecular weight proteins tend to move rapidly through the matrix because they experience less resistance during the time of electrophoresis. As a result, we can see the nearer of the protein band is to the well means that the larger of the molecular size of the protein molecule.
For this part, we will look how SDS page works. Material we need are DC power source, tank, glass plate, spacer, plastic cone, polyacrylamide gel, buffer system, and protein. So let's look how to run the SDS page. To start the procedure, 2 gel are needed in SDS page, which is supporting gel and stacking gel. For casting the gel, 2 glass plates are needed, shorter one and tall one. The shorter will be placed in front and put at the casting frame. After that, place it at the casting stand. Separating gel then will be pipetted at the bottom with pH 8.8. After that, stacking gel will be on top with pH 6.8. And on top of that, wells are formed by using plastic cone. While waiting for the gel polymerize, we have to prepare protein sample by add protein with SDS and a tire reducing agent such as beta-mercaptoethanol. The gel used for SDS is polyacrylamide gel. Beta-mercaptoethanol is functioned to reduce disulfide pitch. After that, heat it into incubator with temperature 95 degrees for a few minutes. After SDS treatment, we can see the charge are uniform because tertiary and secondary structure are completely unfolding. After the gel are solidified, it will be transferred to electrophoresis apparatus. Gel compartment will be added and cone must face to it. Buffer solution will be added properly to avoid any leakage. After that, comb will remove gently and the denatured protein sample will be piped into the wells. Electrode will connect it to the power supply. When the power supply is applied, the negatively charged protein will run through the gel from cathode to anode. The protein will travel through the gel with different velocity. Large molecule more slower than small one. At the end, after running the gel, we will stain it to see the path by using Comasi Blue, a dye to visualize protein. After Comasi staining, the protein band then will be revealed. When we want to confirm the specific protein, the gel then will be undergo Western blot.
SDS page is widely used in biochemistry for the quick and exact separation and subsequent of analysis protein, mostly used in analytical purpose and less for preparative purpose, especially when larger amount of protein are to be isolated. So I'm going to explain to you a few applications of SDS page. Firstly, SDS page is used in Western blotting to determine the specific protein in a mixture of protein. Next, SDS page is also used in post-translational modification of protein that can lead in different relative mobility, such as band shift or a change the binding detection of antibody used in Western blotting, which we can see that the band disappear or the band appear. Lastly, the SDS page is also used in medical diagnostics such as in HIV tests and to evaluate the proteinuria. In HIV tests, the protein are separated by the SDS page and subsequently detected by the Western blotting, which have the specific HIV antibodies that present in a blood serum. Meanwhile, in proteinuria, SDS page will evaluate the level of various serum protein in the urine such as albumin, alpha-2 macroglobulin, and immunoglobulin G. Here are another application list of SDS page, which is used to measure the molecular weight of the molecule, and in peptide mapping, it also used to estimate the size of protein and to estimate the purity of the protein. And lastly, it's also used to compare the polypeptide composition of different structure.